Hello everyone, you are now watching TanyaCikgu live from BAC Flex Studio. My name is Fong and I will help you to answer all the questions regarding biology, SPM for the next 2 hours. Send your question in the comment box or FB Messenger SPM Flex. Thank you. Okay, so for number 1, okay, this soalan from Tan. Okay, can I explain about the process of transmission of nerve impulses along 3 types of neurons and the process of transmission of nerve impulses across synapse? Okay, I think I try to answer your first question. It's about impulse travel along the 3 types of neurons. So of course there are 3 types of neurons there. Number 1 is called the afferent neuron. One more name for afferent neuron is called the sensory neuron. Then for number 2 is called the motor neuron. One more name for motor neuron is called the efferent neuron. And number 3 is called the interneuron. I think I put it this way, I use one reflex action to explain to you the flow of impulse along these 3 neurons. I think it should be better. Okay, let's say for a reflex action, for example, if this one is a very hot surface and my hand touch a very hot object, I will withdraw my hand immediately without thinking. This is actually called a reflex action. And why without involved thinking? Because the whole thing is controlled by the spinal cord. Okay, let's imagine when I first touch a very hot surface here, okay, there is a hot receptor under my skin here. So the hot receptor will be triggered and impulse is sent by the afferent neuron all the way to the spinal cord. Once enter the spinal cord, to be more specific, is the gray matter of the spinal cord, impulse is transmitted to the interneuron in the gray matter of the spinal cord. And then from the interneuron, pass the impulse to the motor neuron, also in the gray matter of the spinal cord, and all the way this impulse is carried by the motor neuron all the way to the, let's say, muscle effector to respond. Maybe I draw for you, it's better. Okay, let's imagine this is the cross-section of our spinal cord. And this, the middle part, is called the gray matter. And then we have also the white matter here. Okay, let's say this part here is the spinal cord. The middle here, this is the gray matter. The outer part is actually the white matter. In BM it's called jirim kelabu and jirim putih. Okay, let's see here with the receptor. We talk about reflex action. We talk about, let's say, heat or the hot receptor, detect stimulus, and impulse is triggered, sent by the afferent neuron all the way to the gray matter until inside here, the gray matter of the spinal cord, then ending like this. And here got one circle and the dot. Okay, this one circle and the dot is a cell body, and this one represent our afferent neuron. Okay, one more name for afferent neuron is called the sensory neuron. Okay, and then from here, in the gray matter, impulse is passed to the interneuron. This one is the interneuron here. Okay, and then pass to the motor neuron all the way up. This one is up, this one is in to the effector. Effector is any part of the body that respond to stimulus. Normally, example here is muscle. We talk about physical response, is about the muscle. If talk about homeostasis, then involve the effector is our hormone, means endocrine gland. Okay, and this one is our motor neuron. One more name for motor neuron is called efferent neuron. Look at this one. This is starting is the afferent neuron, is sensory. This is the efferent neuron. One for the input A, one for the output is E. Okay, I hope that with this diagram can help you understand the flow of the three neurons. Okay, so again, so when you detect anything by your receptor, impulse of course is triggered and is sent by the afferent neuron or the sensory neuron all the way to your spinal cord. They will all the way enter until the gray matter, the inside the region. Then only impulse is transmitted to the interneuron. And then from the interneuron, it will pass to the motor neuron and send all the way out to your body, normally say to your muscle here, to respond. Actually, reflex is to protect your body from serious injury. So basically, reflex is controlled by the spinal cord and not involve any thinking. So it's automatic response. Okay, so for these three neurons, one more time, they all send impulse. They got the same function. Their function is to transmit or to send impulse. Okay, just that for the afferent neuron, send impulse from receptor to the interneuron. From receptor to the interneuron, this one is by the afferent neuron. Then the inter send the impulse from afferent to efferent neuron. Then for the efferent, send impulse from the interneuron all the way out to effector to respond. Okay, yeah. And then after this one, the next part you need to focus is here. The gap here, this gap here between nerve and nerve, the gap is the one that we call synapse. This one. So when impulse all the way travel until the end of one nerve, how to across the synapse to the next neuron, right? So this one is about the synapse, is about the neurotransmitter. Here involved the chemical is called neurotransmitter. Okay, maybe I open another page for you and I explain to you one more time about how impulse across the synapse to the next neuron. Okay, let's say at the top here, this one is the synaptic knob. And then the bottom one, this is the dendritic. The dendritic is really like the antenna one that can receive the impulse. And this is actually the receptor. This one is actually the whole thing here is actually dendritic. And then for this one is receptor. Okay, and then for the top, this part here is called a synaptic knob. Okay, and then of course got the vesicle. Inside got a chemical called neurotransmitter. Okay, and got one more thing you have to focus is here got also mitochondrial, another mitochondrial here. Okay, let's say when impulse all the way from top arrive at the synaptic knob here, how to pass this impulse across the gap here called synapse. Okay, answer is the arrival of impulse at the synaptic knob trigger synaptic vesicle. So the vesicle here is called synaptic vesicle. So trigger the synaptic vesicle to release neurotransmitter into synapse. Here you get 1 point. Next point, then the neurotransmitter, this chemical here is called neurotransmitter. They can actually diffuse across the synapse and bind to the receptor here, means the dendritic. The binding of neurotransmitter to the dendritic or to the receptor here trigger a new impulse generation, means when neurotransmitter bind to this receptor, it will generate a new impulse to continue the impulse flow here. Okay, so one more time, the process of transmission of nerve impulses across the synapse, the main point is actually neurotransmitter. So imagine synapse is just a very narrow gap between two neurons. When impulse flow until the end of one nerve, almost to the next neuron, this is very narrow gap here is called the synapse. So basically these two neurons, like the cable, they are not connected physically. So how to pass the impulse from one impulse, something like the electric, okay? How to pass the impulse from one cable neuron to the next cable across the gap here? Answer is electric cannot jump out from the cable. So answer is through the chemical form called neurotransmitter. Okay, so one more time here, when impulse arrive until the end of the neuron, means it's called the synaptic knob here. The arrival of impulse at the synaptic knob will trigger this part to the vesicle called synaptic vesicle to release the chemical called neurotransmitter into the gap. Then the chemical molecule here called neurotransmitter will all the way diffuse across the gap and bind to the receptor at the dendritic here. So the binding of neurotransmitter to the receptor trigger a new impulse formation to continue the flow here. Is it okay or not? Okay. In case for this type of question come out in your exam and they will ask give example of neurotransmitter. So example for neurotransmitter, try to memorize at least one example. There are three altogether in your syllabus. One is called dopamine, one is called noradrenaline, another one answer is acetylcholine. Okay, I write for you here. For the chemical, got three example. We have dopamine, article. Thank you. Okay, we have dopamine here, number one. Then we have number two, answer is noradrenaline. And number three, answer is acetylcholine. And how to memorize these three, answer is dopamine, start with the D. So it's D. Noradrenaline, start with the N. And the acetylcholine, start with the A. So it's actually DNA. So DNA, there are three chemical here, represent dopamine. N is for noradrenaline, and A is for acetylcholine. So the three, try to memorize at least one. Okay, yeah. When it comes to this type of question, they can ask you also something like application one. Let's say if got chemical, got the drugs, they can bind to the receptor here, block here. Okay, got drugs can block here. What happened to the impulse transmission? Okay, answer is if block 2, let's say got 3 receptor here, if block 2, response will become slower. Because at first you got 3 receptors accept the chemical. Now left only got 1. 2 are blocked by the chemical, the drugs or the poison here. So answer response becomes slower. If all the receptor blocked by the chemical or the drugs, answer is impulse cannot pass through at all. Means no impulse, no response. Okay, okay, I hope that this one can help you understand about the nerve. Okay, so one more time here, the 3 types of neuron. We have the afferent or sensory neuron, interneuron, and then the efferent or the motor neuron. Okay, and how the link I will show you in the diagram for the reflex one, and how impulse transmit across the synapse, and this is the answer. So the main answer is neurotransmitter. Okay, I hope this one can help you understand better and can help you answer better in your exam. Okay, for the next one, also from, okay, same student. Okay, what are the famous type of questions will come out in SPM about chapter of reproduction and coordination. Okay, for chapter reproduction means it's your Form 5 chapter 4. So for Form 5 chapter 4, popular one, okay, let's say we talk about this year. Okay, based on the past year and the past few years, they're already out about the menstrual cycle, talk a lot about menstrual cycle already, and also about the contraceptive method also. So quite some time that not yet come out actually is about the pregnancy. Okay, for about the pregnant mother, about the placenta thing, about the twins formation, about identical twins, the non-identical twins, and also one more special case is the Siamese twins. So for this one, you really need to focus for this year. And then the popular topic, famous one about reproduction, of course, still back to gametogenesis, means about spermatogenesis, and the oogenesis, how to form the sperm, the process meiosis, how to form the ovum, and then you compare these two. Okay, this is number one. And then followed by menstrual cycle, for girl period cycle, about the 4 hormones, the graph, okay, and then is the placenta thing, placenta, talk about placenta. So normally for placenta, they can ask about the function of placenta, for example, okay, and then about the twins formation also, okay, the formation of baby twins, got identical twins, non-identical twins, and also the Siamese twins, okay. And then another one is not so common, is about the plant. From chapter reproduction is double fertilization. So basically there are only few popular one for SPM about chapter reproduction. One more time, gametogenesis. You have to compare girl and also the boy, how to form sperm, how to form ovum, the whole process. Mitosis, meiosis, memorize everything, haploid, diploid. Number 2 is about the menstrual cycle, the 4 hormone in graph form. And then it's about the pregnancy, twins formation. And last one is double fertilization, means for the flower. So based on my personal opinion, okay, my advice is focus more on the pregnancy part, okay, about the twins formation, that part, okay. Then the next one for chapter coordination means actually is the chapter 3, Form 5 chapter 3. Say for coordination one, okay, for this one, okay, just now I already explained, is about the neuron, reflex. Yes, the one's under coordination. Yes, reflex is popular, about the synapse is popular, about the neurotransmitter is popular. Another popular thing, okay, for past 2 years SPM, they come out about kidney also. So kidney is about the urine formation. So already out 2 years for the kidney. So beside kidney, yes, they can ask again one more time for kidney also. So beside kidney, which one you can focus? Kidney is the most difficult one under homeostasis, already out 2 years already. So beside kidney, you can focus on like sugar level, or maybe about the body temperature, how to regulate the temperature, body temperature, the hair erect, all these is under coordination. Okay, then for the next one, okay, about the CFC, okay, for another student. Okay, can I explain about CFC? Okay, so CFC is actually your Form 4 syllabus, chapter 9. Is the last chapter for Form 4 syllabus. It's under the pollution part. So basically CFC, of course, it stands for chlorofluorocarbon. So basically any chemical with chlorine present, with fluorine present, with carbon present in one molecule, that molecule is called CFC, chlorofluorocarbon. Means chlorine, fluorine, and carbon together as one molecule. So it's called CFC. And what CFC can do about here? Of course, CFC is the cooling gas, for example. Let's say when we turn on the aircon, so of course we release the CFC. It's actually in gas form. So this CFC gas will flow all the way to the atmosphere, the layer, the ozone layer there. So when come to the ozone layer, this CFC is very dangerous one. Means this CFC is very bad one. It will actually attack our ozone layer. It will destroy our ozone layer and make our ozone become thinner, thinner, and thinner. So once become thinner and thinner, as we know, ozone layer is to protect us from the UV, from the sunlight. So when the ozone layer become thinner, it means more UV can come to the Earth's surface. So if more UV come to the Earth's surface, any effect here, more UV means more sunlight. Of course, we increase the overall temperature, like global warming, may cause global warming due to the thinning of ozone layer. Yes, thinning of ozone layer can also link to global warming because increase the temperature, more UV. Then the next one is more UV to human can cause skin cancer. Skin cancer mainly caused by actually UV from sunlight. Okay, skin cancer, and also can cause eye cataract, means mata, the eye, then got one layer grow there and block your vision. So basically you're sitting a bit blur one. Eye cataract also caused by UV, high UV. Okay, skin cancer, eye cataract, and can also weaken our immune system. When the temperature is very hot, so basically we don't feel comfortable because the temperature is very high, so enzyme cannot function well. So weaken our immune system. And then for plant, high UV also can destroy the chlorophyll, so plant cannot survive. When plant cannot survive, means animal cannot survive. Okay, so about the CFC, I'm going to explain to you also how this CFC molecule destroy our ozone layer, involve a series of chemical reaction. So let's start now. Okay, so CFC is any chemical molecule with at least one carbon, at least one chlorine, at least also one fluorine here. Okay, of course, for every single carbon atom should be with 4 covalent bond. Let's say here got 2 Cl and then 2 F. Of course, at least one chlorine, at least one chlorine, at least one fluorine, at least one carbon. Then it's called CFC, chlorofluorocarbon. Also can be 3 chlorine and 1 fluorine, or maybe 3 fluorine, 1 chlorine also can. Make sure that total got 4 here. Okay, and then when this is actually gas, CFC, this is actually gas. When this gas all the way flow until the ozone layer, okay, UV from the sunlight are very strong. UV from the sunlight, UV, got the ability to break this chemical bond. When here break already, means the chlorine will come out as individual 1 chlorine atom. The one that attack our ozone layer is actually this guy called chlorine atom. It's actually here. This is the one that attack the ozone layer because chlorine atom is not stable. Based on chemistry, chlorine 1 only is not stable. If Cl2, chlorine molecule, this gas, this one is stable. But when come to here atom, Cl1 only, we normally put here 1 dot. This is called free radical, means it's not stable. When this is not stable at the ozone layer, what they have? Ozone molecule. This is the ozone, O3, this is ozone molecule. So when chlorine atom present there, plus this is not stable, the dot here means free radical is not stable. When it's not stable, it will attack the ozone molecule. And ozone means got O3, got 3 oxygen. This chlorine will take 1 oxygen from the ozone and will form here ClO. This is called chlorine monoxide. Okay, and remain is the O2. This one is actually the oxygen. So chlorine monoxide and oxygen, this one stable, this is also stable. But the main point is this step already destroy 1 ozone molecule, become O2, become ClO here. So basically 1 ozone molecule is destroyed in this step. And then this ClO will further react with the oxygen atom. From where can get this oxygen atom? Answer is from oxygen gas, O2. The O2 got double bond here, same story. The UV from the sunlight, very strong, can break this double bond at the atmosphere layer there, and then here form the chlorine atom, form the chlorine atom also. This chlorine atom actually can react with also oxygen, and all combine O2 plus 1O will become the O3. This is called the ozone molecule. This for ozone layer formation is actually good thing. This is actually good one. It's ozone layer formation. But the point is with the pollution, this ClO present, the chlorine atom might react with the ClO and take the O here, become oxygen stable, and the Cl back to Cl dot again. Means now this is not stable, like back to the starting point. It will repeat the experiment. So basically this Cl dot will actually repeat. The whole thing means destroy another ozone layer. Scientists predict that like this one Cl dot present in ozone layer can repeat this process about 100,000 times. Means can destroy about 100,000 ozone molecules per year. And it's non-stop. So basically it's about repeat the thing and destroy more and more ozone layer. So basically, yes, CFC, this one is cooling gas. Aircon, we need CFC, no choice. But actually now we got the solution for this case already. What we can do, we can replace the CFC with the HCFC. H is stand for hydro. So hydrochlorofluorocarbon. This is environmental friendly product that won't destroy our ozone molecule in the ozone layer. Okay. So CFC one more time is about the thinning of ozone layer. So how this molecule can destroy the ozone, already explained to you just now. Okay. And the effect of course is more UV come to the Earth's surface. So more UV means more sunlight. More sunlight can increase the overall temperature. So it's global warming. Okay. Hope this one can help you understand better about CFC. Normally come out is in essay format. Okay, so for next question, okay, from another student here. Okay, how to analyze diagram of phases of mitosis and meiosis, because you always get wrong in objective. Okay, so this one, okay, what we can do is, number one, you have to familiar with the mitosis first. The four diagram, we have the prophase, we have the metaphase, anaphase, and telophase. So it's PMAT. Okay, this is number one. And then for the meiosis, we have meiosis 1 and we have meiosis 2. It's also PMAT for both pro, meta, ana, telophase. Just that meiosis got 2, we got meiosis 1, right? And then the meiosis 2. So for the meiosis 1, of course, it's called prophase 1. Then metaphase 1, anaphase 1, telophase 1. Meiosis 2, the same thing here, just put the 2 behind, like prophase 2, metaphase 2, and so on. Okay, so about the diagram, okay, very simple one. Number one, you must understand, for meiosis 1 got the special diagram. How special? The chromosome got paired up. Once you see chromosome got paired up, means that one black, one white, they pair up together. This is called homologous chromosome. When it comes to homologous chromosome, must be meiosis 1. Confirm already. Okay, once got homologous chromosome in the diagram, then confirm is meiosis 1. If no pair up, just like one like this, single, the X chromosome, just normal, let's say for prophase example. For prophase, if only one like that, then no pair up, can be mitosis or can be meiosis 2, can be prophase 2, can be just normal prophase. Let's say talk about prophase now. But if got pair up, for the prophase, got pair up, got homologous chromosome form, answer is meiosis 1, prophase 1. Okay, yeah. And then how about, you want to know more about this one? I show you the diagram. We talk about prophase, for example. Prophase, prophase 1 and prophase 2. Okay, for the prophase diagram, let's assume that diploid equals to 4 chromosomes first. So for the prophase, you will see there are 4 chromosomes all together. Yes, already condensed, got 4 chromosomes. And of course, for prophase diagram, the nuclear membrane should be with dotted line, so that going to disappear. he also disappear. We'll focus on the chromosome first. So you can see 1, 2, 3, 4 chromosome. This is actually the prophase because no pair up. But for prophase 1, they will actually pair up. And this pair up process is called synapsis. They will form one pair, one pair. Normally must be the same size. They come together in the nucleus for the prophase. So you can see pair up like this. When you see one pair, this is called homologous chromosomes. So basically, when you see homologous chromosomes pair up like this, confirm is meiosis 1, no other, only meiosis 1, so it's prophase 1. But for prophase 2 is a bit special. Basically, prophase 2 and the prophase mitosis is about the same diagram, but the main point is he only got 2 chromosomes. Okay, one more time here. We assume that for this case, diploid is 4 chromosomes. Here got, yes, 4 chromosomes. You can see 1, 2, 3, 4. And this is actually diploid. For this one, only got 2 chromosomes. Means these 2 chromosomes, this is actually haploid. If you see the haploid form, then it's 2. If diploid form is the normal mitosis. When you see one pair, homologous chromosome, and then answer is meiosis 1. Okay, so I think for exam, popular diagram is about the prophase diagram, metaphase diagram, and also the anaphase diagram. Okay, so it's for the prophase diagram. You see homologous, then it's automatic is 1. No homologous can be 2 or can be just mitosis. So just look at the diagram. Is it diploid or haploid first? Maybe I'll show you one more example. Let's say we talk about the metaphase. This is metaphase. This is metaphase 1. Metaphase 2. When got 1 and 2 is for meiosis. Okay, we talk about metaphase 1. So for metaphase 1, you can see the homologous chromosomes, one pair, homologous chromosomes, they line up at the cell equator. When you draw the diagram, you have to show that the spindle fiber touch the centromere and touch the centriole. Okay, for the metaphase 1, you can see it's one pair, is homologous. And then for the metaphase, it's 1 by 1, single. Got 4 also, but 1 by 1, line up at the cell equator, line up at the middle of the cell. Need to touch middle here, touch, and touch the centriole, touch the centromere, touch the centriole. Must connect them because spindle fiber will pull them to the end one. So we need to connect like this. And then for the metaphase 2, it's also 1 by 1 single, but here only got 2 chromosomes. Why only got 2 chromosomes? Compare metaphase 2 and metaphase, about the same. These two about the same. Also chromosomes 1 by 1, chromosomes 1 by 1, line up at the cell equator. Only for metaphase 1, special case is 1 pair, homologous. But the rest is 1 by 1 here. So you can see this one is 4 chromosome, diploid. Here is 2 chromosome, haploid. Okay, so I give you the summary one more time here. When come to the objective, about the diagram here, when you see got homologous chromosome, automatic must be meiosis 1. No other answer. But when you see the diagram, it's like normal one, it can be like mitosis or can be meiosis 2, where you can see to get answer is count number of chromosome. So if the total number of chromosome equivalent to diploid cell, then it's mitosis. If haploid means half, then it's meiosis 2 already. Or another way is when come to meiosis 2, got one more special case, you can see got crossing over effect or not. The black and white got mixed or not. If got mixed, automatic is meiosis 2 also. Because crossing over only for meiosis. mitosis, no crossing over at all. So I hope that you can really get the point here and really help you in your exam. So it's very popular topic, mitosis meiosis, in your exam. Make sure that you can read the diagram properly and make sure that you choose the right answer. So when you see pair up, homologous, meiosis 1. No pair up, if no crossing over effect at all, is the mitosis. Got crossing over effect is the meiosis 2. And also diploid mitosis, haploid meiosis 2. Okay. Okay, so we come to another question. Okay, Shasha. Oh, nitrogen cycle. Okay, I explain one by one first. So it's about nitrogen cycle number 1. Nitrogen cycle can go to, like, let's say essay, about 10 marks essay. So altogether, there are 5 bacteria you need to memorize. For example, we have something called Rhizobium species that live in the plant nodule, okay, to help the plant to fix nitrogen and give this nitrogen actually to the plant to form protein. It's under the mutualism part, okay. So in return, the plant also gives shelter and food to this bacteria to stay in. So basically, both sides also benefit. So the first bacteria you have to know from this one called nitrogen cycle is called the Rhizobium species. Or maybe I'll give you a big intro first for this cycle. Nitrogen cycle, why we need to study? Okay, answer is actually nitrogen is needed by plant and also by human, by animal to form protein. Want to form protein, we need 4 basic elements. We need the carbon, C. We need the H, hydrogen. We need the O, oxygen. And we need also one more is the N, is nitrogen. Imagine, plant can perform photosynthesis. They can form sugar from water and carbon dioxide. They can form actually sugar. Sugar got CHO. But sugar lack of nitrogen. Okay, and want to form protein, 4 elements must be there. Okay, must be there, these 4 elements. So got CHO from the photosynthesis. If can add in the nitrogen, then only can form protein. Can form protein, then you can form the new cell. Then the plant can grow. So basically, a plant yes also need protein. Okay, to form new cell to grow, they need also protein. Since photosynthesis only provide CH and O, they need also nitrogen. And where to get this nitrogen? Answer is fertilizer. Fertilizer. So actually fertilizer, they also provide nitrogen, provide this nutrient, nitrogen to the plant to form protein. Okay. So about this cycle number 1, I say this nitrogen gas from atmosphere, and then will be first fixed by the leguminous plant. Let's say this is ground, underground. The rhizome got the nodule. Okay, yeah. Inside this nodule, live a type of bacteria is called the Rhizobium species. Okay, this one is to fix nitrogen. Function is to fix nitrogen. Okay, when come to exam, if you can mention this bacteria name, Rhizobium species, you must underline. Then the SP dot, you get 1 mark direct. Function is another one. Okay, so basically this one called Rhizobium species is a bacteria, fixed nitrogen. So you get 2 points altogether. So after this one, let's imagine human can eat plant. So from plant protein become animal protein. And after some time, both will die. Okay, after die, what happened is the decomposer will come to decompose the dead body to become ammonium. Okay, the NH4 plus. Okay, ammonia and then ammonium also. Okay, from here, this bacteria that involved is the decomposer. So the decomposer, this bacteria, you put it saprophytic bacteria. So for saprophytic bacteria, you'll get here 1 point. Function is to decompose the dead body
into ammonium. So decompose is the second point. Decompose dead body into ammonium means return everything the protein to the soil. Here you get another 1 point, 1 point also. Then in the soil under the ground, they got this bacteria is called Nitrosomonas. Nitrosomonas species. They can convert ammonium become the nitrate ion. It's NO2 negative. Okay, yeah. And then from here nitrate, there's another bacteria called nitrobacter. They can actually convert nitrate become the nitrate, the NO3 negative. Look carefully here. One is NO2, one is NO3, one is nitrate is I, one is nitrate is A here. This is the general one, is the fertilizer that can be absorbed by majority land plant. Even by other land plant, the big one, also can absorb this one as the natural fertilizer to form protein. Okay, if got any excess of this ion that cannot be absorbed by the plant, it can be returned to the atmosphere in the gas form by another bacteria. Is called denitrifying bacteria. Okay, so one more time. All together, got few bacteria to know. Number 1 is the Rhizobium species. You can name the bacteria, you'll get 1 point. Function is 1 point. Number 2 is the saprophytic bacteria. Number 2, bacteria, saprophytic, or fungi also can. Function, decompose, 1 point, 1 point. Then Nitrosomonas, number 3. Name this bacteria, underline, SP dot, you get 1 point. Function is to convert, I just say convert, ammonium into nitrate. Function is 1 point, bacteria is 1 point here. Then number 4, Nitrobacter. Same thing, bacteria, function, also say convert. Convert nitrate into nitrate. Okay. Then the last one is number 5, denitrifying bacteria. Convert nitrate to nitrogen gas. Also convert. You get 5 bacteria altogether. So basically for this cycle, yes, you need to put a bit effort to go through the whole thing one round first, because when come to exam, this nitrogen cycle is quite popular also for essay. So in case come out in essay, they will give you the whole diagram. They will give you all the ammonium, the name, the nitrate, nitrate, everything, but they won't give you the bacteria name. So the bacteria name is your answer. What you have to do is memorize the how to spell every single thing, because when come to exam, all the scientific name, one name, you will get 1 mark. So what they want is perfect spelling, underline the name, and then SP dot. That's it. Just one name, you get 1 mark. So must be perfect spelling, underline, and then SP dot, species. Okay, and then the function. So there are 5 bacteria that involved in nitrogen cycle. So got 5 function, maximum 5 bacteria, 5 marks. 5 function here, also 5 marks. So total is 10 marks essay. In case come out in the MCQ for this cycle, maybe they just give you part of the cycle. Like let's say ammonium to nitrate, nitrate to nitrate, that's it. Okay, only one small part only here. So if from ammonium to nitrate, what is the bacteria here called? Nitrosomonas. If from nitrate to nitrate, what's the bacteria here called? Nitrobacter. Okay, so this one is for MCQ. The next one is about, okay, how about niche habitat community population for the differences? Okay, niche means the role, the peranan. Okay, let's say for plant is producer. Okay, and then for the habitat is actually the natural habitat. Like for example, forest or hutan is actually natural habitat for animal, for plant. So for plant, animal, their natural habitat is actually forest, means tempat tinggal mereka. Okay, habitat. Then for the community, okay, I come to population first. Population refer to a group of people, a group of organism of the same species. Let's say human population in PJ. Okay, human population in Kepong, human population in KL. So how many human in KL? Only human. Okay. Then how about if you want to know how many cat in KL? So it's called cat population. How many dog in KL? Dog population. How many human in KL? So human population. So it's one by one, must be same species, you put one group. Human population, dog population, maybe elephant population, for example, cat population. So it's 111. When all these living organism they combine together, let's say okay, human, dog, cat, elephant all combined together, is called a community. Okay, so basically community is all the living thing they join together. Okay, so when all the living thing join together, it's called a community. Okay, so I hope that this one can help you a little bit. So just refresher, population is just a group of the organism of the same species, human, one group, human population. Cat population, dog population, all combined is called a community. Okay, okay, so move on to the next one. Okay, this soalan from Yingyi. Enzyme, why enzyme is used in the production of baby food? Okay, I think this is under the application. Okay, why baby food they need enzyme? Because for baby, okay, their body cannot digest the food like means like adult. So their digestion still a bit slow one. So if let's say we put the enzyme in the food, digest for them. So when they eat, direct is the simple form. Okay, imagine let's say we eat something like rice, is complex carbo. We eat noodle, complex carbo also. So for all this rice, let's say imagine for baby, we don't give baby, let's say 1 month to eat rice. Because rice is a big complex carbo. They need time to digest. Am I right or not? So what we can do is for the baby food, we add the enzyme so they can digest already or hydrolyze already the complex carbo into the simpler form. So the simpler form is a small size molecule. So when baby eat, no need to digest, just direct pass through and direct absorb. So for baby, this is actually very good one. Okay, so one more time, for baby, they need the enzyme. Baby food need the enzyme is for the digestion because they cannot digest like adult first. So they need something like liquid form. It's easier for them to absorb. I hope that this one can help you understand that. Yes, it's under application of enzyme. Okay, next one from this student called Alan. What is centrosome? What is centrosome here? Okay, we have the centromere in the syllabus. We have the central in the syllabus. This one centrosome, I don't think is in our syllabus. Okay, so okay, let's say if they ask about the centromere, then it's about the chromosome part. Let's say chapter 5, form 4, this one chromosome. Okay, and this one, the middle one here, is called the centromere. Okay, let's say we talk about the cell division from the spindle fiber. Is it for one cell? Example, metaphase. This one is called the central. Okay, yeah, so we have the central and the centromere. So this one is centromere. Centromere, and central in our syllabus. This one, centrosome, I don't think is in our SPM syllabus. Okay, so I hope that this is what you are looking for, so can help you a little bit understanding on the chapter cell division. So it's about the DNA part. Okay. Okay, so for the next question, okay, from this student called Kanzim. Okay, so can you explain the effect of overuse of painkiller to the transmission of impulses between neurons? So this actually I think under soalan kebat because it's not in the syllabus thing, but we can explain one. Okay, about the impulse transmission.
Okay, it's about the neurotransmitter thing. So effect of overuse of painkiller. So basically painkiller is like the block the receptor. Just now I said that. Okay, when impulse arrive at one end of the nerve, how to transmit the impulse to the next nerve across the synapse. So basically the gap here is called synapse and the chemical thing is called the neurotransmitter. So receptor here and the dendrite got receptor. So the receptor here once receive the impulse, receive the neurotransmitter, then the impulse can be transmitted to the next neuron. With the painkiller, they will actually block the receptor. So when they block the receptor, answer is yes, you don't feel the pain, right? Okay, but of course it's good thing for you. But the long-term effect is not about this one. The long-term effect is painkiller can destroy, can cause liver damage and also the kidney failure. Means when come to the medical part, painkiller overuse actually will destroy the liver and also our kidney. And about the actual function way, how the painkiller function is block the receptor. So that when you feel the pain cannot send impulse to the brain. So you don't feel the pain. Means from the pain receptor detect the pain already. The impulse should be sent to the brain one. But with the painkiller, they block the receptor. So your impulse cannot pass through. So you don't feel so pain. But for the long-term consumption of painkiller, actually will cause liver damage and also kidney damage. Okay, yeah, so it's not recommended. Okay, it's not recommended to overuse the painkiller. Okay, so we move on to the next one. Okay, Fahmi. Oh, this year Kelantan paper. Okay, is the interaction burung hantu dengan tikus. Okay. Okay, yang pertama ialah tentang namakan hubungan antara tikus dengan burung hantu. Jawapan ialah mangsa pemangsa, prey and predator. Sebab burung hantu dia makan tikus. So it's about prey and predator, mangsa pemangsa. Yang pertama. Lepas tu untuk yang nombor 2, untuk graf dia. Selepas masa X, after the time X, apa yang berlaku ialah terus sambung, still like what up down, both also like up and down. Because this one is must achieve the dynamic equilibrium. So katakan kalau burung hantu dia naik, tikus akan mula turun, tikus turun, dia pun turun like this. Macam gelombang. Okay, yeah, for these two. Because it's under mangsa pemangsa one. Then explain your answer. Okay. Untuk graf itu, sekali lagi ya, katakan selepas time X, selepas tu X bagi burung hantu dekat bawah sini kan dia akan naik, turun, naik, turun. For burung hantu, lepas tu bagi tikus pula, dia ke atas kan. Untuk tikus tikus lah begini, macam gelombang. This one is for mangsa pemangsa punya graf. Okay. Lepas tu terangkan apabila populasi, katakan populasi tikus meningkat, ada banyak makanan bagi burung hantu. Jadi untuk dia ada makanan, dia boleh tumbuh. Katakan tikus meningkat kan, tikus meningkat, diikuti dengan burung hantu pun akan meningkat di sini. Meningkat, ini pun meningkat. Kenapa? Sebab tikus merupakan makanan, food. Bagi burung hantu ada makanan, dia boleh tumbuh. Lepas tu apabila populasi tikus ini turun, dia pun kurang makanan, dia pun turun sama-sama. Boleh faham? Jadi naik turun begini, macam gelombang punya. This one is for the prey and predator graph. Lepas tu untuk B. Ramalkan apa yang berlaku jika pokok padi diserang oleh serangga perosak. Kalau padi, bayangkan padi dimakan oleh tikus. Kalau tikus terdapat makanan,
tikus mati, maksudnya populasi tikus akan turun kan. So apabila populasi tikus turun, burung hantu pun akan turun juga. Sebab tanpa makanan. Sebab paddy, okay, jawapan itu adalah pengeluar, producer. So when there's no producer, you cannot get a primary consumer to survive. Tanpa pengeluar, tikus tu tak ada makanan. Tanpa makanan, tikus akan mati. Jadi populasi tikus akan turun. When ini turun, of course bagi burung hantu pun akan turun sama-sama. So it's like the chain effect here. Producer turun, primary consumer pun turun, then secondary consumer pun turun juga. Okay, ya. So kena beritahu satu demi satu, one by one. Okay, so from the paddy, pokok paddy merupakan makanan bagi tikus yang pertama. So apabila pokok padi diserang oleh serangga perosak, dikurangkan, jawapan ialah populasi tikus akan turun sebab tanpa makanan. Lepas tu populasi burung hantu pun akan turun juga. Okey ya. Okey, yang last kali bahagian C. Okey. Kaedah memelihara burung hantu lebih baik berbanding penggunaan pestiside. Adakah anda bersetuju? Berikan alasan anda. Okey. Inilah soalan kebat lah. Kelantan paper. This year paper. Fikir dulu. Okey. Untuk yang ni, okey, memelihara burung hantu lebih baik. Okey. Sebab pestiside itu, dia akan bunuh tikus. Jadi akan menyebabkan populasi dia akan turun. Kalau kamu bagi dia makanan di mana dia boleh hidup dengan lebih baik, dibanding dengan dia ambil tikus sahaja. Okay, this one is soalan yang perlukan pandangan kamu. Adakah anda setuju? Okay, jadi jawapannya lah ya. Okay, alasannya lah, so the burung hantu punya populasi kamu boleh kawal. Jawapannya lah ya. Okey ya. So ini daripada soalan Kelantan 2019. Soalan yang bagus ya. More to soalan kebat here. Okay. So yes, your SPM for past few years banyak soalan kebat. You kena cuba linkkan apa yang kamu boleh faham daripada buku teks ke bahagian soalan. Okay. Hope that this one can help you. Okay, next one. Okay, for another student. Okay, boleh terangkan bagaimana penyusunan kromosom homolog menyumbang kepada variasi suatu spesies. Okay, ini ialah bahagian form 5, bab 6, bab variasi. Last kali punya. Okay, bagaimana penyusunan kromosom homolog menyumbang kepada variasi. Okay, untuk menghasilkan variasi, okay, adanya dua faktor. Satu ialah faktor genetik, yang kedua ialah faktor persekitaran. Di mana untuk SPM, kita hanya fokus pada faktor genetik sahaja. Dan untuk faktor genetik, ada beberapa jawapan yang boleh menyumbang kepada variasi. Yang pertama ialah pindah silang. Okay, pindah silang untuk Prophase 1. Yang kedua ialah yang ni, penyusunan kromosom homolog secara random. Okey, boleh menyumbang kepada variasi suatu spesies. Okey, saya cuba gunakan diagram. Ini kamu boleh gunakan Mendel yang kedua untuk terangkan. Okey, contoh adanya 4 kromosom. Ini sepasang kromosom homolog. Another pasang kromosom homolog. Okay, katakan sini adanya jin bagi jin tinggi, ini jin pendek. Lepas tu katakan bahagian bawah, ini jin boleh gulung lidah, ini tak boleh gulung lidah. Metaphase. Penyusunan kromosom homolog kat sini. This is the first possible result. Bayangkan, kalau ini T besar kat sebelah sini, G besar means dua rupa dominan di sebelah yang sama. Bayangkan semasa meiosis 1, ini dua pisah. Akhirnya sampai gamete terbentuk, gamete sebelah sini akan terima T besar iaitu tinggi, gene untuk tinggi dan gene boleh gulung lidah. Okey. Sebelah sini pula akan dapat ialah small t untuk pendek dan small g tak boleh gulung lidah. Katakan untuk lelaki, inilah sperma. Ini pun sperma juga. Of course, ini dua-dua, big T, big G, dominant. Ini small t, small g, dua rupa resif, tak sama DNA kan? Yang pertama. Okay. Kemungkinan penyusunan kromosom yang kedua, okey, bayangkan sama juga kromosom homolog susun pada sata khatulistiwa. Okay, the second possible answer. Okay, bayangkan untuk T adalah sama. Untuk T besar, dominant. Ini T untuk yang pendek punya, small t. Sama. Ini adalah sama. Okay, sama. Katakan yang bawah tu dia terbalik. G besar kat sebelah sini. This is a small g. Nampak tak? Tadi dengan sekarang ada beza. Itu G sudah pun terbalik. Sudah pun tukar. Di mana mestinya ini dua kromosom homolog disusun terbalik. Jadi sini big G, sini small g kan. So if like this, when here adanya pemisahan kromosom homolog, gamete sebelah sini akan terima satu big T tinggi, satu small g tak boleh gulung lidah. Gamete, katakan sperma. Ini pula akan terima satu small t untuk pendek dan satu big G untuk yang boleh gulung lidah. Okay, cuba bandingkan empat jawapan. Big T, big G pertama. Small t, small g kedua. Big T, small g nombor 3. And small t, big G nombor 4. Jadi untuk ini 4, semua pun tak sama DNA. Kalau tak sama DNA, maksudnya boleh menghasilkan variasi. So dari segi penyusunan kromosom homolog, boleh menghasilkan, katakan penyusunan kromosom homolog yang secara random punya, dia boleh memberikan jawapan ialah menghasilkan gamete yang berbeza DNA. Kalau berbeza DNA, maksudnya adanya variasi. Okey, sekali lagi penyusunan kromosom homologous, penyusunan kromosom homolog pada sata khatulistiwa semasa metafasa 1. Dia boleh susun begini ataupun dia boleh terbalik. Secara random punya. Begini boleh, begini pun boleh, random kan. So kalau dia random, keluar punya gamete, DNA pun tak sama. Kalau DNA tak sama, boleh menghasilkan variasi. Ya, untuk manusia, setiap satu gamete yang kita hasilkan melalui meiosis. Setiap satu gamete pun tak sama. Means every single sperm, dia memang unik punya. Boleh faham? Ovum pun sama juga. So in this world, everyone muka pun tak ada sama. Semua tak sama DNA punya. Disebabkan adanya yang pertama pindah silang, yang kedua ialah penyusunan kromosom homolog secara random pada sata khatulistiwa semasa metafasa 1. Okay, I hope this one can help you understand slightly better. Okay, ataupun kamu boleh rujuk pada hukum Mendel nombor 2. Dia memang sama punya. Okay, kenapa berdapat 4 jenis gamete yang berbeza? Inilah jawapan. Disebabkan penyusunan kromosom homolog secara random pada sata khatulistiwa semasa metafasa 1. Okey Okey, cuba yang next one sekarang. Okey, daripada Shah. Okey, cikgu boleh bandingkan perbezaan antara pencernaan di mulut dan di perut. Okey. Ini short answer saja. Okey, untuk pencernaan di mulut adanya 2. Yang pertama ialah pencernaan fizikal. Maksud kamu kunyah tu makanan. Okey, you memecahkan makanan from bigger size jadi smaller size kan. Itulah fizikal punya. From bigger size become smaller size. Kamu kunyah. Okey, lepas tu yang kedua pencernaan dari segi kimia.
Jawapan melibatkan enzim. Apa enzim ada di mulut boleh mencernahkan makanan? Jawapan ialah amylase. Amylase Leo. Ada alio kita adanya amylase Leo. Amylase Leo hanya boleh mencernahkan kanji sahaja. Okey, hanya kanji. Boleh tak amylase mencernahkan lemak? Tak boleh. Boleh tak amylase mencernahkan protein? Pun tak boleh. Amylase hanya boleh mencernahkan kanji kepada mautosa. So di mulut dari segi pencernaan kimia hanya ada satu jawapan, iaitulah amylase Leo mencernahkan kanji, katakan nasi ke, mi ke, semua pun adanya kanji, roti ada kanji kan. So mencernahkan kanji kepada mautosa. Dan untuk kanji, okay, just for information, kanji merupakan kompleks karbo kan. Okay, kompleks karbo dia tak manis. Kalau tukar ke mautosa, mautosa ialah disaccharida. Dia manis, rasanya manis. Maksudnya kalau kamu makan nasi, kamu kunyang betul-betul, katakan 15 saat ke, okey, dan air liur kamu pun keluar, boleh mencernahkan kanji, tukar kepada mautosa, di mana kamu akan rasa nasi itu adalah manis. Boleh faham? Kalau kamu makan nasi, masuk dan telan, masuk telan, kunyang 2 kali, telan saja, jadi tak akan ditukar kepada mautosa kan. You tak akan rasa nasi itu manis. Boleh faham? So you kunyang betul-betul, air liur masuk, mencernahkan kanji kepada mautosa, mautosa adalah manis. Okey, di mulut yang ni. Okey, lepas tu yang kedua, di bahagian perut. Okey, pencernaan di perut, bayangkan perut artinya bergerakan peristalsis kan. Itu dikira pencernaan fizikal. Okey, tapi untuk exam biasanya kita fokus pada pencernaan kimia. So pencernaan secara kimia di perut hanya ada 2 jawapan. Sana perut ada 2 enzim sahaja. Yang pertama dinamakan ialah pepsin. Pepsin boleh mencernahkan protein. Okey, ya. Pepsin boleh mencernahkan protein kepada polipeptide. Maksudnya mulut hanya boleh mencernahkan kanji, iaitu karbo sahaja, kepada mautosa. Di perut hanya boleh mencernahkan protein sahaja. Boleh tak? Boleh tak perut mencernahkan kanji? Tak boleh. Sebab perut dia boleh merembeskan, namanya ialah jus gastrik kan. Jus gastrik dia adanya hydrochloric acid, so memberikan keadaan asid. Okey. Yang kedua dia adanya enzim. Enzim ada 2 enzim. Satu ialah pepsin. Yang kedua ialah renin. Jadi untuk pepsin ialah untuk mencernahkan protein, okey, kepada polipeptida. Itu saja. Lepas tu polipeptida akan masuk ke bahagian duodenum untuk sambung pencernaan dia. Lepas tu yang kedua, renin. Enzim renin, di mana renin dia ialah untuk coagulate. Dia bukan mencernahkan. It's like coagulate, bagi dia ke bentuk yang lebih pepejah. Casinogen kepada casein. Ataupun beritahu, okay, renin menukarkan casinogen kepada casein. Apa itu ialah casinogen? Susu protein. Okay, protein susu, milk protein. So basically, untuk di perut, dia hanya mencernahkan semua tentang protein sahaja. Sekali lagi ya, mulut satu jawapan, amylase Leo mencernahkan kanji kepada mautosa. Okey, yang pertama, of course kunyang, pencernaan fizikal. Lepas tu di perut, peristalsis, pencernaan fizikal. Pencernaan kimia ada 2 enzim, pepsin dan renin. Okey, how to memorize? Senang saja. Untuk pepsin dan renin, pepsin dia P kan? Okey, pepsin dia P ya. Renin adalah A. Perkataan perut so P, pepsin. A, renin. So di perut adanya perut PA. Pepsin renin, perut. Okey ya. Jadi pepsin lah untuk mencernahkan protein yang biasa daripada daging ke ikan ke, protein kepada polipeptida. Lepas tu untuk renin ialah menukarkan casinogen kepada casein. Inilah protein susu. Milk protein, casinogen. Apakah beza casinogen dengan casein? Jawapannya adalah casinogen larut
bentuk yang cecair punya. Inilah tidak larut. Something like semi-solid, tak larut. Okay, ya. Okay, so kalau sampai perut saja, lepas tu sampai sinilah. Untuk perut, 2 jawapan saja. Okay, lepas tu mulut, only one answer is the amylase. Okay, ya. Hope that this one can help you understand better about chapter 6, pencernaan. Okay, next one. We come to another one. Tihani belajar. Okay. Teacher, can you explain more about spermatogenesis and oogenesis? Okay, can. Spermatogenesis and oogenesis. Okay, spermatogenesis when compared to the oogenesis, you must know, number one, spermatogenesis is to produce sperm for boy. Okay, occurs in testis. Even to make it more specific, answer is testis. Spermatogenesis occurs in which part of the testis? Answer is seminiferous tubule of testis for spermatogenesis. And then for the oogenesis, means the ovum production. For the oogenesis here, answer is occurs in ovary. Okay, so for the difference more, okay, number one is produce sperm, one is produce ovum. Number one. Number two, for spermatogenesis, location is seminiferous tubule in the testis. For oogenesis, in general, just say in ovary. Actually, oogenesis occurs in ovary and also the fallopian tube, but it's not only happen when it comes to pregnant that time. Okay, yeah, because for the oogenesis is slightly more complicated. For oogenesis and spermatogenesis, both also involve actually meiosis 1 and also meiosis 2. But for girl, when they perform the meiosis 1 and 2, the cell divide unevenly into two, one bigger and one smaller. So the big one after meiosis 1 is called the egg cell. It's called the egg cell, the real egg cell. So when come to the ovulation, this egg cell bursts from ovary and come out into the fallopian tube. It's the egg cell. If got sperm enter the fallopian tube, penetrate the egg cell, then the egg cell can perform meiosis 2 and form ovum in the fallopian tube. Okay, yeah. So basically, if put it in general way, yes, oogenesis occur in ovary, finished. But to be specific, is in ovary and the fallopian tube. But normal is just ovary. Okay, about the product. Spermatogenesis, one diploid cell can form 4 functional cells, means 4 sperm cells. But for the oogenesis, from one diploid cell can only form 1 ovum. So 4 functional unit, spermatogenesis, 4 sperm cell. But for the oogenesis, yes, got 4 haploid cells, but functional only 1, only got 1 ovum. Another 3 called polar bodies, they are non-functional cell, so only got 1 functional cell and 4 functional cell for spermatogenesis. Oogenesis only got 1. And then for the next point is spermatogenesis, okay, can complete the whole process by their own. But oogenesis normally is if got sperm present, they then only can complete the whole oogenesis. Okay. Another difference is for spermatogenesis occurs in boy 1, they start to perform spermatogenesis after they reach puberty. For boy around age of let's say 11 to 14, okay, then they start to produce sperm from starting until ending. The whole process about 3 months. But for girls, since fetus already start oogenesis, but of course until halfway stop, then reach puberty, continue a little bit. When got sperm enter, complete the whole process. Okay, maybe I show you in the diagram form to let you understand better. So we start with the spermatogenesis. Okay, for spermatogenesis, start with a diploid cell. It's called a primordial germ cell. And then from here got repeated mitosis. So it's mitosis, so it's still diploid, it's mitosis. And then from here can form the one is called spermatogonium. Spermatogonium. And then if you grow to the bigger size, matured one. This is the growth phase, like the G1S and the G2, and now it's called primary spermatocyte. And then from here, I start meiosis 1, split into 2. These two same size, both also called secondary spermatocyte. And then followed by meiosis 2, split one more time, all also same size. Total got 4 haploid spermatic. Spermatic. And then go through, okay, for boy got one special processor. It's called the cell differentiation. Cell differentiation from basal cell and form the actual sperm cell. Sperm cell. Sperm cell. One more name for sperm cell is called spermatozoa. This is a scientific name for sperm. It's called spermatozoa. So for boy, from top to bottom, the whole process is about 3 months. Okay, so start from the spermatogonium, then go to the primary spermatocyte, secondary spermatocyte, spermatic, and spermatozoa. So meiosis is actually here. Here start meiosis 1. So when they start meiosis 1, the bottom one, all haploid, all N haploid. All N haploid. Before meiosis 1, the top here all diploid. All 2N. 2N, 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 2N. Bottom here all haploid. Okay, so for boy. And for the girl, oogenesis. Okay, let's say if I change the title to oogenesis here. It's about the same name for girl. Also start with the primordial germ cell, same answer. Just that this one called spermatogonium, right? The spermato, you change to oo. So become oogonium. It's the same answer. Spermatogonium for sperma. Now oo is the oogenesis. Okay, yeah. Grow to the big size is called primary. Change to oocyte. Oocyte. Secondary for girl, secondary oocyte. From spermatocyte, change to oocyte. Cyte is the same. Cyte means cell. Okay? Cyte means cell. Like erythrocyte means red blood cell. Leukocyte. Cyte is actually cell. Okay, yeah. And then, okay, for girl, oogenesis, the main difference is when it comes to the meiosis 1 here, for girl, should be one bigger. And this one will become the smaller. If for girl, okay, here will become smaller in size. This one is bigger. Meiosis 1, here bigger. Haploid, here smaller. The small one is called polar body. It's non-functional one. Polar body or first polar body. Then this one is a secondary oocyte. This is the one that we call egg cell. Let's say secondary oocyte. This is the one that we call egg cell. So during ovulation, is this egg cell enter the fallopian tube. If got sperm enter the body, come to the fallopian tube, let's say for the sperm cell want to penetrate this egg cell in the fallopian tube, then only can trigger meiosis 2 to occur for girl. And when come to meiosis 2 for girl, it's the same story. Also got uneven cell division. Also one bigger, one smaller. All this also small size. The small one all non-functional. Polar bodies, no function. The functional only one is the ovum. Okay, so one we compare one more time between spermatogenesis and the oogenesis. Beside the product sperm and ovum, location testis and ovary. Okay, for spermatogenesis got cell differentiation. Oogenesis not involve cell differentiation. Okay, and then spermatogenesis, one diploid can form 4 functional cells, 4 sperm cells. But for the oogenesis, the 4 haploid, only one is functional, only one ovum,
another 3 just polar bodies, non-functional. Okay, yeah. If you want to compare about the differences, I think should be quite enough info already about the location, about the product, boil angle, okay, and also the uneven distribution of the cell during meiosis 1 and the meiosis 2. Okay. Okay, next one from this belajar. Okay, Arun. Okay, apakah subtopik penting yang perlu study bagi pemerhatian lebih sikit untuk study dalam 2 minggu? Oh, maksudnya kamu nak tahu apakah ramaran maksudnya. Okey. Ya, saya ada sikit idea juga. Okey, beginilah. Untuk bio yang pertama, you kena tahu cara mereka set soalan. Untuk katakan 100%, the whole paper 100%. Katakan okey, normally yang form 4 punya dia kan set lebih kurang 55 hingga 60%, means the whole paper 100% kan. 50 hingga 50 ke atas hingga 60% semua form 4 syllabus. Make it 60-40 lah, lebih senang. 60% ialah form 4, 40% ialah form 5. Okay, you cuba fikir. Kalau 60% form 4, maksudnya saya study form 4 lebih, saya pun sudah pun boleh pass. Okay, let's say saya punya goal ialah I want to get credit C ke atas. Saya hanya belajar form 4 saja. Katakan form 5 saya tak boleh faham. Semua form 4 pun tak mau belajar. Saya hanya belajar form 4 saja. Saya boleh tak dapat C? Jawapan boleh. Jawapan ialah boleh ya. Sebab form 4 60%. Form 5 40% saja. Okey, kalau cuba tengok yang kertas lepas punya, benar keluar hanya form 4 saja boleh mencapai 78 markah. So it's about 78%. So 78% not only credit is also about A already. Okay, A or A minus hanya form 4 saja. Tambah sikit form 5, of course is A or A plus. Boleh faham? Tapi kamu kena tahu untuk form 4 adanya banyak topik. Katakan daripada chapter 2 sampai chapter 9 ada banyak. Okey. Kita mula daripada chapter 2. Untuk chapter 2 tentang sel organelle, ini biasanya kamu perlu fokus untuk soalan struktur. Yang basic animal cell ke, plant cell ke, okey. Amoeba ke, bab 2 punya, biasanya itu bahagian struktur. Lepas tu untuk yang bab 3, ada 2 bahagian you kena fokus. Yang pertama ialah fluid mosaic model. Dia punya struktur, label dan fungsi sekali. Yang kedua, of course, ialah osmosis. Okey ya, yang hypertonic. Lepas tu untuk bab enzim, of course is lock and key. The enzyme characteristic. Okey. Lepas tu untuk bab 5, cell division, mitosis, meiosis, itu perlu tahu. Lukis, terangkan setiap proses. Katakan untuk profasa 1, terangkan 2 markah. You kena beritahu tentang sinapsis ada berlaku dan pindah silang pun berlaku. Contoh, okey. Dah beritahu ialah daripada bab 2 hingga bab 5 form 4 punya 2, 3, 4, 5 sampai pembahagian sel. Kalau you belajar ini 4 bab, jawapan ialah minimum 2 soalan struktur confirm every year punya. Okey, maksudnya belajar 2, 3, 4, 5 confirm ialah 24 markah boleh keluar. Okey, itulah minimum. Kalau maksimum benar keluar ini 4 chapter kat 1 year, 4 soalan struktur. Bab 2, 1 struktur. Bab 3, 1 struktur. Bab 4, 1 struktur. Bab 5, 1 soalan struktur. Semua pun ialah 12 markah. Itu 48 markah. Sudah pas. But normally, minimum pun ada 2 struktur. Ataupun 2 struktur campur 1 essay. Itulah 44 markah. Pun boleh pas. Boleh faham? Okey, untuk essay apa yang boleh keluar? Contoh yang lebih penting ialah osmosis. Ataupun ialah pembentukan enzim. Okey, the enzyme formation punya. Okey. Lepas tu untuk bab 6, okey. Dari segi pencernaan, itu perlu tahu pencernaan dan juga penyerapan makanan dan juga asimilasi. Okey. Lepas tu lagi satu yang agak susah punya kerana keluar ialah tentang tindak balas cahaya dan tindak balas gelap. Itu tumbuhan punya. Okey ya. Lepas tu untuk bab 8, respirasi aerob dan anaerob.
manusia dan yeast. Okey. Lepas tu untuk yang bab 8 dan 9, ini dua, itu ekosistem kan? Ini biasanya kamu kena belajar untuk esei. Ini untuk esei punya. Okey, ekosistem mainly untuk esei saja. Popular topik bagi exam termasuk tadi CFC punya penipisan ozon layer, ya, boleh keluar esei. Lepas tu deforestation sudah pun keluar 2 kali. Okey, tahun lepas. Lepas tu pemanasan global. Then itu 3R recycle punya, reuse, recycle that one. Dan colonization and succession bagi kawasan Paya-Pakau. Okey. Lepas tu ialah dari segi yang basic, katakan rantai makanan pun kena tahu juga. Okey, untuk bab ekosistem ini biasanya keluar untuk esei dan ada juga di kertas 1. Jadi pun perlu belajar. Ini untuk form 4 sahaja. Okey ya. Lepas tu untuk yang form 5 punya, topik yang penting katakan ialah jantung yang pertama. Okey, jantung, struktur jantung, penairan darah melalui jantung bagaimana. Lepas tu yang kedua, ini agak susah sikit, ialah sistem limfa. Okey, sistem limfa. So sistem limfa can link to sistem pertahanan badan. Okey ya. Lepas tu untuk yang bab 2 punya form 5 tentang locomotion and support, hanya satu konsep penting is the antagonistic muscle. When one muscle contract, one muscle relax. Itu saja. And of course a bit about tulang belakang. Okey ya. Lepas tu untuk bab 3 lebih ke bahagian reflex dan juga homeostasis. Okey, contoh how to control body temperature, terangkan semua sekali, bentuk esei ataupun struktur. Yang reproductive punya bab about pregnancy punya twins formation, sekali lagi ya, twins formation, and then about the spermatogenesis, oogenesis, kita haid. Okey, dan bab 5, okey, Menda memang dia penting. Dia hanya formula pengiraan macam matematik. Pengiraan genetik punya. Menda itu popular. Normally Menda boleh keluar untuk struktur ataupun esei. Bab variasi yang kamu perlu tahu ialah dari segi apakah beza variasi selanjar dan variasi tak selanjar, dan juga mutasi. Okey. So overall kena tengok kamu punya goal. Kalau kamu nak katakan kamu hanya nak pass saja, sebenarnya advice ialah you hanya perlu fokus pada form 4, you confirm boleh pass. Tapi katakan you mau dapatkan A. Dapatkan A untuk bio, dia kena belajar semua sekali. Sebab A, especially lah A plus, kena belajar semua sekali. Okay ya. Okay, next one. Pada belajar ni. Okay, Marvin. 10 markah. Menstrual cycle. Okay, berdasarkan graph, based on the graph, explain the relationship between pituitary hormone and ovarian hormone with the changes in ovary and the endometrium. Okay, ini 10 markah punya essay tentang kita haid. Okay, kita kena bahagikan begini kepada beberapa bahagian. Yang pertama, katakan dari hari pertama hingga hari yang kelima, apa yang berlaku. Okay, contoh. Dari hari pertama hingga hari kelima. Okay, from day 1 to day 5. Okay, what happened to the endometrium wall? The endometrium wall breakdown. Maksudnya ialah period. Okay, masalah period lah. Day 1 to day 5. The wall here breakdown means having period. Okay, and then at the same time, the pituitary gland will here start to secrete a hormone called follicle stimulating hormone. So from day 1 to day 5, this hormone increases. From day 1 to 5, FSH hormone increases. You can look at the graph also. Day 1 to day 5, the hormone increase is the first graph, and here is the hormone Q. The Q is the FSH. Okay, so day 1 to day 5, this hormone increase. Why this hormone increase? Answer is to stimulate follicle development in ovary. Okay, so the follicle development in ovary, you can look at the last graph just above the endometrium wall there. Okay, so let's say you can mention what happened one by one. Day 1 to day 5, again, day 1 to day 5 here. Endometrium here break down, means you can see the endometrium become thinner and thinner. This actually having period is 1 point. Okay, and then the pituitary gland, they talk about the hormone Q, means it's the FSH here, increases, you get also 1 point. And then what is the function here? It will actually stimulate the follicle development in ovary, is also 1 point here. Okay, lepas tu lah day 6 to day 13. Just before ovulation, apa yang berlaku? Okay, untuk FSH ini akan turun. Means the hormone Q akan turun. Pada masa yang sama, hormone X ialah estrogen from the ovary. From the ovary, here we secrete actually more hormone X, means estrogen. X, based on the graph ialah X. Ini turun, ini akan naik. Okay. Kenapa begini? Sebab the info actually from day 1 to day 5 sini. From day 1 to day 5, FSH ini keluar bertujuan untuk merangsangkan perkembangan follicle, is for the follicle development here. So when follicle here develop in the ovary, follicle in the ovary is the one that secrete the estrogen. So as the follicle grow bigger and bigger, estrogen become more and more. When estrogen start to increase, this hormone can inhibit the FSH. Merencat dia. Means when this one increase, automatic FSH will drop. Because this hormone stop the FSH production. Okay, so from day 6 to day 13, you can mention, okay, FSH drop, ovary secrete estrogen, here increase, and estrogen is to repair endometrium. Repair endometrium after menstruation. Means after period. Okay, so here also 1 point. Okay, I go through these few points for you first because it's 10 marks essay. I break into 2 parts. So day 1 to day 5, the endometrium breakdown. As you can see, the graph is all the way down. So breakdown, you get actually here 1 point, means having period. And then for number 2, the pituitary gland secrete the FSH, means it's the hormone Q based on your graph there. And this hormone is to stimulate the follicle development in ovary. Okay. And then the follicle growing in the ovary will actually secrete the estrogen to repair the endometrium after period. Estrogen also can inhibit the FSH. So the FSH, the graph dropped from day 6 to day 13. So already got 6 points altogether. So we continue, already got 6 points here. Okay, then on day 14, special here, the day of ovulation. Okay, so for the day 14 here, you have to mention is about the hormone P already. Hormone P is the LH, luteinizing hormone, is secreted by the pituitary gland. And of course, this gland secrete the LH, okay, luteinizing hormone here. You get also 1 point. This is the hormone that stimulate the ovulation to occur on day 14. Also 1 point here. Besides can stimulate ovulation, luteinizing hormone also stimulate the corpus luteum formation. Okay, corpus luteum formation in ovary, of course, in ovary, and also stimulate the corpus luteum to secrete progesterone. Okay, so one more time. So on day 14, the special case here, of course, you have to mention the main thing is about the ovulation. This is the main event here on day 14. But who stimulate the ovulation to occur? LH. Who secrete LH? Pituitary gland here. Okay, and then from here, LH got three functions. Number one, ovulation, 1 point. Number two, stimulate corpus luteum formation, 1 point. And stimulate corpus luteum to secrete progesterone, also 1 point. Here also 1 point. So got 4 points here. Okay, just now already got 6 points. Here got 4 points. Yes, enough to get 10 marks already. But of course, this essay can go to about easily more than 13, 14 marks in the marking scheme. So after the day 14, what happened next from day 15 to day, let's say 25, okay, before the next period. From day 14 to day 25, answer is corpus luteum will continue to grow, okay, continue to develop and keep on producing progesterone. Progesterone function is to maintain the thickness of the endometrium or further thickening. So from the graph after day 14, you can see number one, got the thing is the corpus luteum there, formation. Then it's the corpus luteum that secrete the hormone Y, progesterone. And hormone Y here is to maintain the thickness of the endometrium. So about the same thickness or slightly thicker or further thickening the endometrium until day 25. Okay, so 3 points. Already 3 points here. And then when it comes to day 25, let's say if no pregnant, no fertilization, after day 25, you can see the graph in the ovary one, the corpus luteum will become smaller and smaller, right? So corpus luteum start to degenerate. Okay, degenerate, smaller and smaller. Okay, 1 point from day 25 onwards. Okay, corpus luteum degenerate after day 25 of the cycle. Progesterone hormone also drop because it's the corpus luteum that produce the progesterone one. So when this corpus luteum becomes smaller and smaller, also produce less and less progesterone. So with lesser progesterone, no one maintain the thickness of the lining anymore. So without the hormone progesterone, the endometrial lining will start to break down and come out as period for the next cycle. So normally about day 25, the hormones start to drop. The endometrium start to break from the crack. So at this point also, where hormonal imbalance occur, there's another topic for the PMS. The premenstrual syndrome is actually come from the hormone drop. Suddenly the hormone Y, progesterone, drop about 2-3 days before the next period come. Okay, yeah. But for this essay, I think it should be here enough to get 10 points. Okay, so this one can really help you. Okay, for the next one, okay, from Chia. Okay, greenhouse effect. Okay, for the greenhouse effect here, also more to the essay. So about greenhouse effect, you can explain about the punca dia, means the factor that can cause the greenhouse effect to occur. The main answer is the release of carbon dioxide. So anything, any human activities that we do can release or increase the CO2 gas, also can mention in the essay. For example, like burning or combustion of fossil fuel, burning of forest burning, and also deforestation from the vehicle, burn the petrol. So all the burning can release carbon dioxide, and also deforestation also can increase the CO2. Okay, so all this, yes, once increase CO2 here, so the CO2 gas can actually trap the heat. Okay, when it comes to greenhouse effect, you can also explain about how the thing trap the heat. For example, first is the sunlight. Okay, will all the way pass through the atmosphere layer, come to the Earth's surface, and this sunlight is called the UV, ultraviolet. So some of this light at first will filter by the ozone layer first. Okay, reflect first to the space, and some will pass through and come to the Earth's surface. So when come to the Earth's surface, of course, plant will absorb the sunlight. for photosynthesis, absorbed by the Earth's surface, but some will be reflected back to the space, right? Okay, the point is when you want to reflect this heat back to the space, this heat now is called the IR, the infrared. The sunlight direct from the sun to the Earth's surface is called UV, very strong heat wave. Okay, very strong energy. When the reflect one with lesser heat, right? Lesser heat is called IR, infrared. So this infrared, okay, the heat is actually trapped by the CO2, the greenhouse gases. So when they trap the heat here, this heat will be reflected back to the surface. So overall, we increase the temperature a little bit. Increase a little bit at first is okay. But when more and more CO2 gas trap the heat, we increase the temperature to very high level until the term is called the global warming. So from the global warming, what happened next? Answer is very hot temperature, of course, climatic change. Okay, suhu berubah. Okay, the climate, okay, change become very hot, and when become very hot, we also affect the wind direction one. Okay, and also from the greenhouse effect can cause melting of polar ice. Okay, melting of polar ice, and from there can rise the sea level, can cause flood as well. So all this effect from the greenhouse effect. So from greenhouse effect, normally try to link to the global warming. Then you can have more point to explain. Okay, I repeat. For this type of essay, number one, you have to know the factor. What caused greenhouse effect? Okay, answer is CO2. That's the main one, greenhouse gases. And what human activities can release the CO2 gas? Burning, forest burning. Okay, and then compassion, deforestation. Okay, this one, all this can cause the CO2 to increase. And then about explain the process, the CO2 gas, how to trap the heat to increase the overall temperature. Then from there, you can link to the global warming. Okay, actually, what is the main difference between greenhouse effect and global warming? Okay, let's say if the CO2 increase a lot, let's say only in KL, for example, only in KL or only in Malaysia. So only one area that happened, the temperature increase that caused by the heat trapped by the CO2, only one particular area is called greenhouse effect. Okay, but when happen to the whole world temperature increase at the same time, it's called the global warming. So only one area, suhu meningkat, temperature increase, become very hot here, is called greenhouse effect, one area. But when it comes to the whole world, then it's called global warming. Okay, yeah, that's the difference. Of course, from greenhouse effect, you can link to the global warming, so they have more point to explain for essay. Okay, I hope that this one can really help you. Okay, we go to the next one. Hira, teacher, can you explain about the role of hormones in the menstrual cycle? Okay, so it's one more timer that we just go through just now, the 4 hormones, the menstrual cycle thing. So, okay, there are 4 hormones involved in the menstrual cycle. So, number 1 is the FSH. Number 2 is the estrogen. Number 3 is the LH. Number 4, answer is progesterone. So there are 4 hormones that involve, and of course they also involve in certain sequence one. Sequence is number 1 must be the FSH first. Number 2 must be the estrogen. Then number 3, answer is the LH on day 14, and number 4 is the progesterone after day 14. Okay, you have to break into like period like day 1 to day 5. Normally FSH hormone is secreted by the pituitary gland. This is number 1, and the function here is to stimulate the follicle development. Or you can say FSH is to stimulate Graafian follicle development. You can say development or formation in ovary. So this is the FSH. Then for number 2 is estrogen. Estrogen, the function for this case menstrual cycle, estrogen is the girl sex hormone. Estrogen is to repair the uterus lining or the endometrium. So keyword is repair. You can say repair or you can say rebuild. Another one is thicken the uterus lining after period, after menstruation. Okay, So this one for estrogen. Number 3 is the LH. So LH got 3 functions altogether. One is to stimulate ovulation. Number 1 is ovulation. And then number 2, answer is corpus luteum formation. Number 3 is stimulate corpus luteum to secrete actually progesterone. So got 3 functions. For LH is the main one. Okay, again, ovulation is stimulated by LH. Corpus luteum formation also by LH. Stimulate corpus luteum to secrete the progesterone is also by LH. So there are 3 functions altogether for LH. Then when it comes to the last one, progesterone, the main function is to maintain the thickness of the endometrium after ovulation. So basically, progesterone is to replace the estrogen to further thickening the endometrium. Okay, so I repeat lah. There are 4 hormones. FSH, stimulate follicle development or graphene follicle development, number 1. Estrogen, repair the uterus lining after period. LH, main answer is ovulation, and then also stimulate the corpus luteum formation from the follicle cell and stimulate the corpus luteum to secrete progesterone, the last hormone. So progesterone is to maintain the thickness or further thickening the endometrium until day 25. Okay, yeah, I hope that yes, this hormone explanation can really help you understand better. Okay, then we come to the next one. Okay. Okay, daripada belajar ini, okay, tentang pencernaan ruminant untuk lembu dan juga untuk rabbit. Okay, ruminant and rodent. Okay, like this. Bagi pencernaan, okay, ruminant, katakan ialah lembu ke, kambing ke, okay. Then the rodent is about katakan anak ke, rabbit. Okay, dua-dua pun special sebab mereka boleh mencernahkan fiber. Okay, boleh mencernahkan cellulose. Dan manusia tak boleh. Okay, sebab dalam badan mereka adanya satu jenis bakteria, ialah bakteria symbiosis. Itulah bakteria yang bagus. Di mana bakteria ini bagi rumen di perut mereka. Di perut pertama dan perut yang kedua. Untuk lembu adanya 4 perut. Ruang yang pertama dinamakan ialah rumen. Yang kedua ialah rumen, reticulum. Reticulum, omasome, abomasome. Okay, begini ya. Rumen untuk lembu, katakan untuk lembu. Kambing pun boleh juga bagi ruminant. Adanya bagi perut dia adanya 4 ruang. Ini A, dia O, A. First A ialah rumen. Yang kedua ialah reticulum. BM jadikan K reticulum. Lepas tu adanya omasome. Dan abomasome. Okey, begini ya. Bagi lembu, 2 perut yang pertama dalam sini adanya bakteria symbiosis. Itulah bakteria yang bagus. Mereka hidup di 2 perut ini, rumen, reticulum, dan bakteria ini adalah bagus. Dia boleh menghasilkan enzim cellulase, di mana boleh mencernahkan selulosa kepada glukosa. Okey ya, sekali lagi ya. Bagi lembu ruminant, perut dia adanya 1, 2, 3, 4 ruang, 4 perut. Di mana untuk perut pertama dan kedua dalam adanya bakteria symbiosis yang boleh merembes enzim cellulase yang boleh mencernahkan selulosa kepada glukosa. Ini untuk lembu. Manusia ada tak cellulase? Kita tak ada punya. Lembu ada. Anak pun ada, tapi untuk anak special case. Untuk anak, dia adanya di bahagian sikum dia. Untuk anak di bahagian sikum dia. Sikum dia adanya bakteria yang sama, fungsi semua pun sama untuk bahagian anak. So basically lembu dan anak dua-dua pun adanya bakteria ini, adanya enzim ini mencernahkan selulosa kepada glukosa. Ini selulosa ialah dinding sel. It's actually fiber. Boleh faham? Okay. Yang biasalah untuk lembu di perut. Untuk anak berlaku di sikum. Itu beza dia. Okay ya. Jadi ini dua ya, perlu tahu juga untuk exam dia memang penting. Okay. Jadi main point lah adanya bacteria symbiosis, satu di perut, satu di sikum dia. Yang kedua boleh merembeskan enzim ialah cellulase yang boleh mencernahkan selulosa kepada glukosa. Okey. Itu saja untuk yang ni. Lain-lain tu semua macam manusia juga. Okay, lepas tu ke next one. Daripada belajar Arun. Okey, soalan dia, enzim jelas jenis protein dalam struktur tertier. Enzim merupakan protein globule yang padat dan berbentuk 3 dimension seperti A ya. Itu A ialah ialah struktur tertier, means the level 3. Lepas tu untuk yang B punya menunjukkan enzim telah berubah bentuk dan tapak aktif sekali. Mengapakah keadaan enzim di 2.2B boleh berlaku? Jawapan ialah ternya asli. Kalau untuk kes begini, bayangkan untuk katakan yang A itu adalah original punya. Dia dalam bentuk protein, globule, padat, 3D, itulah tertier kan. Dan ke bentuk yang seperti B, kemungkinan jawapan ialah berubahan suhu. Suhu meningkat sampai enzim ternya asli. Sebab pada suhu yang tinggi, you boleh bayangkan, ini semua adalah asid amino. Mereka gabung sekali melalui ikatan kimia. Suhu yang tinggi boleh pecahkan ikatan kimia tersebut dan menyebabkan bentuk itu berubah. Jadi enzim di bahagian B itu berubah bentuk dan juga berubah tapak aktifnya sebab suhu yang tinggi boleh menyebabkan enzim ternya asli. So jawapan ialah ternya asli. Dan kenapa suhu tinggi? Yang pertama, suhu tinggi maksudnya kalau melebihi 45 darjah Celsius ke atas bagi manusia punya, enzim akan mula ternya asli. Kalau melebihi 60 degree Celsius, ternya asli sepenuhnya. So normally kalau untuk enzim ialah suhu 60 dan ke atas, enzim ternya asli. Suhu tinggi di mana ikatan kimia itu pecah. Jadi bentuk dia pun ubah juga. Yang kedua ialah tentang asid dan base. Selain suhu boleh menyebabkan enzim ternya asli, yang kedua jawapan ialah asid dan base. Katakan untuk katakan protein ini dia perlu keadaan neutral. Kalau ada terlalu banyak asid berlebihan atau alkaline berlebihan, di mana dia pun akan ternya asli juga. Jadi dua kemungkinan, satu ialah suhu tinggi, yang kedua asid ataupun base. pH dia berubah. Hope this can help you. It's about ternya asli saja. For the next one, from Tan. What is the difference between neuron without the E and the neuron with the E? Answer is, if you look at the English textbook in Malaysia biology, normally English, the writing part got E behind. The without E one, you look at the BN version for the word neuron is without E one. Again, with E is actually English. Without the E is BM. But if you look at online, with the E is the UK English. Without the E is the US English. But when come to Malaysia, because our English, we follow UK English, so our spelling for the neuron got E. We follow UK spelling. If follow US spelling, then of course it's without the E. BM without the E also. Neuron.
So basically, most of the thing that we can get online, sometimes they will show neuron without the E, means the source is from the US, the link. Okay, with the E, UK English. Malaysia will follow UK English. Without E is for the BM already. But when it comes to exam, don't worry, because with or without the E is still correct. Because we accept both BM and English when it comes to exam, so no issue one. Okay, both also can use. Okay. So overall is okay. For the final one is let you know what you need to revise one more time. Okay, if let's say you are good at the ecosystem part, okay, for something like deforestation, about the CFC, about the greenhouse effect, you can prepare this one for the essay. Okay, let's say you say that, oh, I don't like to study ecosystem at all. About the environment, I got no interest. I don't want to study ecosystem. Can I still pass the paper? Of course, answer is yes. Because the ecosystem normally come out is actually in the essay. And essay, you boleh pilih kan? Okay, let's say got 4 essay. Normally one essay is about ecosystem. Then got another 3 essay. But as a student, you just need to mention, need to answer any 2 essay only. No need to answer all the 4 essay. You just answer any 2 essay only. Without ecosystem, I still can get another 3 essay, right? I still can choose any 2 from here. Okay, yeah. And some students say that, okay, how about if I don't want to study all about plants, tumbuhan, saya tak mau belajar tumbuhan, saya tak suka tumbuhan, I want to study only human and also animal. Can I pass or not? Yes. Normally plants is about 20 to 30% max from the total paper. So even without the plants, you still can pass. Okay, but still the same thing here. You want to get A plus. Okay, you must study everything. Okay, and then the basic thing like chapter 2, 3, 4, 5 is very basic and very easy to score. I hope that this one will help you also. Okay, and then one more thing is about the paper 3. Okay, I think paper 3 we seldom mention about paper 3. So make sure that you know our biology format. Only got 2 experiments, section A and the section B. Okay, only A and B here. So the section A is 33 marks standard, and section B is about the report writing, 70 marks. Okay, some student maybe about the report is not so good. So what you can do within these few weeks, you try to get more past year paper, look at the marking scheme, especially the way they give you the mark. Okay, yeah, so that can strengthen your knowledge about how to answer the technique to answer paper 3. Okay, and then for Form 5 syllabus, the popular one still chapter 1, transport, about the heart, for example, about the blood clotting defense part, okay, lymphatic system, and number 2 is about chapter 3, coordination, chapter 1 and chapter 3, popular for paper 2, okay, coordination and response. Okay, because for this one, why it's so popular? Because this chapter 3 is considered big chapter. So all the big chapter normally will come out in the paper 2. Like chapter 1 is big chapter. Okay, chapter 3 is also big chapter. So the big chapter confirm will come because got many subtopic inside. Okay, and then for the Mendel also popular. Of course, chapter reproduction also yes. And normally chapter Mendel and version they can link together one as one. Okay, or maybe just separate. Okay, so overall, I hope that my this TanyaCikgu can really help you to prepare for your SPM. Okay, so for next week we won't have any slot for TanyaCikgu because we will be celebrating Deepavali. So happy Deepavali! I wish to all who celebrate Deepavali. And TanyaCikgu will be back on Friday, 1st of November with Sejarah. Thank you.